Hemoglobin. We know it as a protein in red blood cells, but how is it synthesized, and what gene is essential for its production as an end product? I chose to investigate the HBB gene, also known as the hemoglobin gene, to gain a deeper understanding of the importance of this protein. Starting in its early stages, HBB is located on chromosome 11, specifically the short arm of the chromosome. Chromosomes in eukaryotic cells are found in the nucleus. An essential process that chromosomes undergo is its opening, allowing for replication and translation. In this process, DNA unwinds and recruits specific proteins, enabling replication to begin at the designated origins of replication, further allowing initiator proteins to separate the DNA strands. Upon further investigation into this process, I learned that helicase, an enzyme that unwinds and separates the two strands of double-stranded nucleic acid, opens the DNA helix, creating a replication fork during the replication process. Furthermore, during transcription, RNA polymerase separates the strands, enabling the reading of DNA's template strand. With this, single-stranded binding proteins help keep the DNA strands apart during replication, proving vital to this process. Moving into DNA replication, we're introduced to how HBB replicates in erythroid cells compared to non-erythroid cells. In erythroid cells, which are the most common type of blood cells responsible for oxygen delivery throughout the body, the HBB gene replicates early in the S phase. The S phase stands for the synthesis phase of mitosis, where DNA replication occurs and the cell duplicates, creating identical copies of each DNA molecule. This process ensures each daughter cell has a complete set of chromosomes when the cell divides. Conversely, when discussing HBB in non-erythroid cells, we discover it replicates later. The overall process consists of 3 steps: initiation, elongation, and termination. Starting with initiation, this step begins at the origins of replication, where helicase unwinds and separates DNA. Next, in elongation, DNA polymerase adds complementary deoxyribonucleotides to each strand in the 5' to 3' direction. Then, in termination, the sequence ends when the replication forks meet. The replication process copies the entire chromosome, including the HBB gene, before cell division. Next, when discussing transcription, we find that it occurs in the nucleus, producing pre-mRNA. The HBB gene is transcribed by RNA polymerase II, regulated by the LCR and various other transcription factors. LCR stands for the locus control region, which is the regulatory region upstream of the beta-globin gene cluster. Diving deeper, beta-globin is a subunit of hemoglobin that the gene HBB codes for, helping control the expression of genes within the cluster. Furthermore, during the initiation phase of transcription, RNA polymerase binds to the promoter upstream of the HBB gene using only the template strand read from 3' to 5'. Next, in the elongation phase, RNA polymerase synthesizes a complementary RNA strand in the 5' to 3' direction using ribonucleotides. The RNA strand is antiparallel to the template strand. Lastly, upon termination, RNA polymerase reaches the terminator sequence, releasing the pre-mRNA. In transcription, there are a few modifications to consider. A 5' cap is added, poly A tail is added to the 3' end, introns are removed, and spliceosomes join exons. These modifications protect mRNA, facilitate its exit from the nucleus, and ensure it encodes the correct protein. Moreover, translation of HBB occurs in the cytoplasm, either on free ribosomes or ribosomes attached to the rough ER. In initiation, a small ribosomal subunit binds to the 5' cap of the mRNA, scanning for the start codon being AUG. Next, the matching tRNA, carrying methionine, the codon for which is AUG, binds via its anticodon, and the large ribosomal subunit attaches, forming a complete ribosome. Furthermore, in elongation, the ribosome reads the mRNA codons. At the same time, tRNA molecules bring specific amino acids based on their codons, and peptide bonds form between amino acids, creating a polypeptide chain. Lastly, upon termination, a stop codon is reached and the ribosome disassembles, releasing the completed beta-globin polypeptide. In addition, when discussing post-translational modifications, the following occurs. The beta-globin chain folds into its tertiary structure, and it can be processed further in the Golgi apparatus if it is synthesized on the rough endoplasmic reticulum. The folded beta-globin then joins alpha-globin chains and heme groups to form functional hemoglobin. These modifications enable the protein to fold and, if necessary, modify it while allowing it to interact with other parts to form hemoglobin in the end. Ultimately, when discussing the gene and its overall purpose, the final gene product of HBB is the beta-globin protein. 2 beta-globin and 2 alpha-globin chains combine to form hemoglobin, the protein in red blood cells responsible for carrying oxygen from the lungs to body tissues and transporting carbon dioxide back to the lungs. This process is essential to cellular respiration, a vital aspect of human health and well-being. In summary, the HBB gene is crucial for synthesizing the beta-globin component of hemoglobin, which plays an important role in oxygen transportation within red blood cells.